histone modifier, and DNA1 you know DNA modifier, DNA methylation mainly. So little bit talk about epigenetics. So otherwise it will not be I'm totally convincing to you guys, and I always speak when I come to this department of Sambhalpur University. I would like to talk about to the students the basics of epigenetics. So what is this? Epi means beyond or above. जब normal को छोड़ के जो जाता है, it is epi means whatever is your gene is there, genetic concept essential for your all functional entity, whatever is happening in the cellular system, metabolism, whatever you call, different kinds of morphologic cell shape, etc., everything. And when genes are regulated properly, you are in good shape, what we call normal health pattern. If it is deregulated, then trouble comes. Now, if you look into this picture, DNA and histone complexes into the total structural features, DNA 2 nanometer in diameter, then nucleosome comes, histone octamers, DNA wraps 1.7 times, you are getting the nucleosome 10 diameter, the nucleosomes complex comes 30 nanometer diameter, and ultimately it is folding to 300 and 700, and at the G1 state of chromatid it is 1400. So the fold pattern it looks like that one, DNA thread, then 4 histones, then whatever you see bits on a string as you have read in typical molecular cell biology book, and then you will see that short range internucleosomal interaction by interhistone H1 or its variant H5. They ultimately help you to fold into chromosomal fiber, and long range fiber interaction ultimately wraps it into the G1 state. So now you are seeing that their DNA and histones they are sleeping very nicely. But all the time if they sleep, who will do their functions? So sleeping must be broken, and it is broken by different kinds of modifications. The basic thing is that in the DNA backbone, sugar phosphate chain is there. Phosphates are negatively charged, and in your histone proteins there are positive chain. Lysine and arginines are there. When histone proteins fold to octamer, it generally protrude the positive charge outside of the cricket ball-like structure as you observe. So these positive charges are utilized by different modifications, mostly methylation and acetylation. You see also, sir, we have observed and we have read in books typically that phosphorylation is also done. Phosphorylation does it, but for differential folding patterns. Some portions are open, other portions when DNA is replicating, certain other portions are needs to be folded. That is newly, that is daughter strands are folded. Okay, so if you look at the basic things, this is all I need not explain. Now chromatin modifications. It will come in the form of DNA methylation. If you look here, this is cytosine. 1, 2, 3, 4, 5th and 6th. 5th carbon of cytosine is methylated. And you see CG, CG, CG or ATCG, CG. But cytosines are very rich in any sequence of DNA, is generally found in promoter regions of many genes, not all genes, in promoter regions of many genes. If it is methylated, then what happens is that methyl binding homoprotein proteins will bound to it and ultimately blocks the entry of the transcriptional factors, ribosome, RNA polymerase, etc. So now if you look at this is DNA methylation, and if you look at the histones H3, H4, H2A, H2B, and you will be positive. But for a single gene, all these type of modifications, as I am showing here, are not required. Certain features according to the cell signaling and the environmental complex, the genes generally are expressed. Typically, there is a correlation, what we call epigenetic signal. After a signal is a transmembrane pathway. Signal is here, enter into the nucleus, and then cyclin D is expressed. Cyclin D gene is expressed in such a way that need not be for other gene, albumin. So here the typical features, as I already explained, the positive charges of lysine, arginine, and phosphorylation also occasionally occurs, and there are various other modifications. So here now genetics, all of you know the study of the genes and how they generated, and epigenetics is the study of how the genes are expressed precisely or remain repressed. Another point joins with that one, RNA interference. All of you may heard about microRNA, various type of siRNA, shRNA. These are not giving us protein, we call non-coding RNA. So these RNA also interfere with cellular processes. Precisely, some of these, although these are a cytosolic event, the repression of the messenger RNA, but some of these are also observed when we analyze the chromatin. The certain microRNA are also participating.